is Joanna Lemley, and I'm a wetland ecologist with the Colorado Natural Heritage Program at Colorado State University. The following presentation will introduce key concepts in the Field Protocol for Lentic, Riparian, and Wetland Systems, the official protocol of the Lentic AIM program. You can follow along in sections 1 and 2 of the draft protocol, starting on page 10. Wetland and riparian areas are transitional between terrestrial or upland environments and aquatic environments like lakes, rivers, and streams. They have high ecological value because they cover a small fraction of the landscape, less than 1% of BLM lands across the conterminous United States. Yet they support high biodiversity and provide numerous other ecological services. Riparian and wetland areas help maintain clean and abundant water within watersheds. They store soil carbon, provide recreational opportunities, have cultural and historical value, and support livestock production. Given their importance in the Western landscape, there is a need for consistent monitoring of riparian and wetland resources across public lands. While BLM lands are generally known as sagebrush country, these lands contain a surprising diversity of riparian and wetland resources. Each of these photos represents a wetland on BLM public lands. And where there is water in a dry landscape, we all flock to it. That includes recreationists, wildlife, and livestock. To address the need for a riparian and wetland monitoring program, the Field Protocol for Lentic, Riparian, and Wetland Systems was developed through an interdisciplinary collaboration between the BLM's National Operations Center, the Colorado Natural Heritage Program at Colorado State University, and the U.S. Forest Service. The protocol draws on several existing protocols developed by the BLM and outside agencies, including the terrestrial and Lentic AIM protocols, BLM's Multiple Indicator Monitoring, or MIM protocol, and EPA's National Wetland Condition Assessment Protocol. It is the official protocol for BLM's Lentic AIM program and joins the family of AIM technical resources. The goal of the Lentic AIM program and the Field Protocol for Lentic and Riparian and Wetland Systems is to provide a standard way to monitor wetland and riparian resources on public lands in order to inform policy, planning, and state and federal regulation. Data collected using this protocol can be used to evaluate the effectiveness of land management actions on maintaining or improving the chemical, physical, and biological integrity of riparian and wetland resources. The data can be used to determine the condition, track trends, and measure annual use of riparian and wetland systems. The protocol is applicable at multiple spatial scales, from fine scale, including site-specific pasture or allotment scales, to broad scales, such as ecoregional, state, or national scales. The protocol and intended uses follow the principles outlined in the BLM AIM strategy and the BLM National Aquatic Resources Framework. Both documents are shown here on the slide. Resource monitoring is an essential component of adaptive management. Monitoring begins with identifying clear management questions, which in turn lead to monitoring objectives that specify when, where, and how monitoring data are collected. The process of selecting sample sites is called a sample design. There are two primary types of sample designs. In random sample designs, sites are randomly selected from all wetland and riparian areas within a specific geographic area. Every member of the target population has a known chance of being selected. Results from random sample designs can be extrapolated to provide a statistically valid estimate of condition and trends across an entire population or study area with known levels of precision and accuracy. In targeted sample designs, sites are hand-selected to address a specific management question, often due to management concern or recent or upcoming management action. Targeted monitoring is appropriate for site-specific evaluations of treatment effectiveness or specific areas of concern, and can be used to document a reference condition, establish a repeat monitoring area, monitor known habitat of a rare plant or animal species, or track changes that result from management actions like grazing or restoration. The Field Protocol for Lentic, Riparian, and Wetland Systems is intended for vegetated riparian and wetland areas, which are sometimes referred to as lentic areas. These include wet and mesic meadows, marshes, seeps and springs, peatlands, including fens and bogs, playas, vernal pools, prairie potholes, kettle ponds, vegetated drainage way, swales, riparian shrublands, sometimes called cars, riparian forests, oxbows, floodplain complexes, floodplains in general, margins of ponds, lakes, and reservoirs. Although not specifically developed for shallow marine or estuarine areas, the methods could be applied in those environments with some testing. There are three critical concepts included in the definition of applicable ecosystems: riparian areas, wetlands, and lentic areas. Those will be explored further in a separate presentation. This slide illustrates some of the more common types of riparian and wetland areas sampled with the protocol, including wet meadows, marshes, fens, vegetated drainage ways, riparian shrublands, and riparian forests. The two types in the lower right can also be described as floodplains. Riparian and wetland areas are characterized by the presence of plant available water above or within the rooting zone caused by inundation, which is flooding by surface water, or soil saturation caused by a high groundwater table. Several different definitions are used for both riparian and wetland areas at the federal and local levels. As I mentioned, we will explore those definitions in a separate video presentation. For the purpose of the protocol, riparian and wetland areas are defined as areas dominated by hydrophytic, water-loving plant species, including those rated as obligate, facultative wetland, and facultative by the U.S. Army Corps of Engineers National Wetland Plant List, wetland indicator statuses. The table on this slide, which is found on page 13 of the protocol, explains the 5 different wetland indicator status ratings. To further bound the environments appropriate for sampling within the protocol, page 15 of the protocol contains a list of criteria that must be met. To be sampleable with this protocol, a site must have at least 10% canopy cover of perennial vegetation. This may exclude playas and other temporary wetlands that lack perennial vegetation. This criterion is included because the core indicators of the protocol are vegetation-based, and data collected in an unvegetated area would not be as meaningful. Within existing canopy cover, the site must be dominated by hydrophytic vegetation, obligate, fac-wet, or facultative species, under typical growing season conditions without recent disturbance events, like a significant or severe flooding, wildfire, or extreme and prolonged drought. Hydrology within the site must be dominated by lentic surface or groundwater, which is slow-moving or still water beyond the immediate banks of a river or stream channel. The following video presentation will further explain the differences between lentic and lotic hydrology. If surface water is present, the monitoring plot must have no more than 10% cover of water deeper than 50 centimeters or 20 inches during the growing season. This criterion does not exclude an entire riparian or wetland area if only a portion has deep water. It merely excludes that portion with deep water. The line point intercept method is very difficult to carry out in deep water, so we have kept the maximum depth at 50 centimeters. Lastly, the site must be large enough to accommodate 75 meters of total transect length in 3 25-meter transects spaced at least 5 meters apart. The methods are not effective for riparian and wetland areas that are too small to accommodate 3 full 25-meter transects. There is not one minimum area threshold, as there are various configurations that can accommodate 75 meters of transect length, including sites that are very narrow, but more than 75 meters long. A separate presentation will detail the various plot layouts included in the protocol. The methods within this protocol are divided into core, contingent, and annual use methods, covariates, and photographs, as described on page 15 and listed in Table 2 on page 17. Core methods are relevant across many different ecosystems and have widespread cross-program applicability. They are required for all monitoring. Contingent methods have the same cross-program applicability, but are only measured where applicable to specific management objectives. Annual use methods are specific to monitoring annual use management activities related to grazing or browse by ungulates and/or human activities. And covariates are measures that characterize the site for classification, stratification, or to determine potential natural conditions. There are 4 core methods in the protocol: species inventory, line point intercept, and vegetation height, litter, and water depth, which were all adapted from the terrestrial AIM methods. Woody structure was adapted from 2 separate methods in the multiple indicator monitoring, or MIM protocol. Those 2 methods are woody species height class and woody riparian species age class. Contingent methods in the protocol include hummocks and water quality. The hummock method was newly developed for this protocol, and the water quality method was adapted from Lentic AIM. Annual use methods include soil alteration, stubble height, and riparian woody species use, which were all adapted from the multiple indicator monitoring, or MIM protocol, and older BLM technical references on annual use monitoring. Covariates help describe the sites being sampled and include photo points, basic descriptive information like elevation, slope, and aspect, which are collected following the terrestrial AIM methods. Classification, hydrology, and soil profile descriptions are all specific to riparian and wetland environments and follow methods outlined in EPA's National Wetland Condition Assessment. Lastly, data on natural and human disturbances are collected. The specific methods used have been adapted from multiple sources, including the EPA, other AIM programs, and NatureServe. The final few slides in this presentation cover elements of Section 2 of the protocol, titled How to Use this Protocol, which starts on page 19. The first element is the standard monitoring plot. Data for all methods and covariates are collected within a defined area surrounding or near a specific sample location, which is a set of spatial coordinates selected in advance using an appropriate sample design, as explained earlier. The sample location, sometimes called a sample point, should be located within or near a riparian or wetland site. The defined sampling area is called the monitoring plot. Data collected within this protocol represent conditions within the monitoring plot, not necessarily across the entire riparian or wetland site. For small riparian or wetland sites, the plot may encompass the entire site. But for large riparian or wetland sites, the plot will only represent a portion of the site. When several monitoring plots are randomly distributed across the landscape, the data will represent the range of conditions within riparian and wetland areas across that landscape. For local or targeted studies of large riparian or wetland complexes, more than one monitoring plot may be needed to adequately represent the range of conditions across the targeted site. The standard plot configuration is the spoke layout. The spoke layout is a 30-meter radius circle that encompasses approximately 0.3 hectares or 0.7 acres in area. The spoke layout is often, but not always, centered on the sample location. The spoke layout includes 3 25-meter transects spaced 120 degrees apart. Each transect starts 5 meters from the plot center. The spoke layout should be used whenever the site can accommodate a 30-meter radius circle. For sites that are too small or narrow to accommodate a spoke layout, the protocol contains 4 alternate layouts in Section 4: transverse layout, diagonal layout, linear layout, and a mixed layout that incorporates elements of multiple layouts. Each layout shares one fundamental characteristic: 3 25-meter transects spaced at least 5 meters apart and evenly spread across the monitoring plot. Each method within the protocol can be carried out independently, but in practice, a suite of methods is carried out during the same site visit. Appendix C on page 160 includes time estimates for each method. Figure 3 on page 21, and included here, illustrates the recommended order of completing all methods. The recommended order assumes a 3-person crew, with 1 crew member leading the vegetation data collection, another crew member leading the soils and water data collection, and a 3rd crew member supporting the others. For some crews, these roles will remain consistent throughout the field season. But other crews may want to change roles at different sites to build skills in all components of the protocol. I will not go through the flowchart in detail because it is hard to understand how the pieces fit together before learning each method. But it is important to see that there is a recommended flow that maximizes efficiency of data collection at each site. While this flow is recommended, site conditions will dictate which methods are used in the actual order of sampling. For instance, not all sites will have surface water, woody vegetation, or hummocks. Given the diversity of riparian and wetland systems, sampling will be different at each site visited. This last slide in the presentation covers timing of data collection. The optimal time to sample core methods is when vegetation is most identifiable, annual soil disturbance is minimal, and water levels have receded. In the Western United States, the optimal time is typically May to September. In contrast, the optimal time to sample annual use methods is during the use period or immediately following livestock grazing or other activities. Practitioners should evaluate the purpose and use of the data to determine when to sample. In some cases, more than one sample visit may be necessary, one to collect core methods and a supplemental visit later in the season to collect annual use data. This concludes the presentation introducing the Lentic AIM program and the field protocol for Lentic, riparian, and wetland systems. Thank you for listening.